Okay. Since you have time, you can go ahead to update your toolbox. I remember last time the app didn't work, but now this should be okay. Yeah, I was looking at the vote, and now I decide to do a presentation on May 6th, exam on the May 8th, so we can finish off everything on that in that week. So this will be the time, and we are going to be in that room on the Tuesday from 10 to 12 to do the presentation, each of you 10 minutes or 12 minutes. The exam will be online, so if you don't want to show up in this room, that will be Thursday on that day. So you can work from home, and I will send you a link so you start from 10 and then finish of your submit your answer using a Google form before 10:55. Then that's it. You don't have to be here on that day. If you want to be here, it's okay. I will be here. Just make sure. Yep.
For the exam, how do you suggest we best prepare for it?
Well, you got to familiar with the PowerPoint slides, so all the questions should be from there. If you think from a examiner perspective, how can I formulate the questions? Basically, I have to prepare a question, then we have 4 answers, and only one of them is correct. So if you look at the PowerPoint slides, there are not too many of those very clear situation. You can have 1 question and 4 answers and only 1 correct. And so just, yeah, and also it's open book, so you can just keep the slides beside you and then work on those questions. Yeah. Yeah, so I'm just updating my toolbox to the latest version. There's several minor improvement, the debugging. And last time the app didn't work, so hopefully this time I think it should work. So the latest version is 4.9. This will be the latest version before you finish. This app should be here and should be updated. And that's where the place you should be able to click and get this. So today we are going to go through graph and networks. So as I said, we always start from X matrix. So this is a cell, gene, cells, you know this. And then we will see how can we construct networks from there. So if you include all the genes, say this could be 8,000 genes. And then your network will be 8,000 by 8,000. So that's your graph. So we are going to do a pre-selection because this is a huge network. We probably will construct the 2000 by 2000 gene regulatory network. So you are going to select the genes is most informative. So and then you build a relatively small network, so it's easy to interpret for the downstream. In our example here, I may select only 500 genes as the example. So we build a small network, and then we can see, visualize it maybe, and then those centrality measures, metric. So that's what we try, and then see whether others we can do compare between two networks and something else. Pseudo knockout, for example, we can try. So that's what we are going to go through using the interface here. So before that, I'm going to show you, so this step you need to build adjacent, so this network is adjacent net, a DJ matrix. So that's what we learned from lecture. So we need to prepare adjacent matrix, which is a network itself. You can either visualize it or not, but this is essential. So we are going to play on in the MATLAB to see if you have adjacent matrix, what we can do next. So we start from here right now, okay? And then I will show you if you have a real matrix of expression, how can you get this? Now we are going to do adjacent matrix, a small one. You can see, okay, there's a whatever. I just made it up. So we are going to type in adjacent matrix in MATLAB, and then visualize it, and show you some examples to do downstream first, okay? So now I need to build adjacent matrix. So I just type it in. If we have, say, 5 nodes, let's just draw a network. Example, 1, 2, 3, 4, 5. If you have 5 network nodes, so 1, 2, 3, 4, 5. I see, okay, 1 and 3 is linked, 1 and 4 is linked, 4 and 5 linked. Let's just do this. Okay, so so if I gave you this graph, this network, translate this into adjacent matrix and type that in there using on your mind. So the first one, there's no self loop. So 1, 1, the position 1, 1 should be 0. And 1, 2 should be 0. 1, 3 should be 1, 1, 4. So you just have to construct this 1, 2 is not. So 1, 3 is 1, 1, 4 is 1. 1, 5 is linked. So this is my for the first row, right? The second, my 2 and 2 is not linked to anything else, just but 3. So for my second row, I will have a 2. Okay. for my node number 3, number 3 is 1 and 2. Okay, so row number 3, 1 and 2 is linked. My row number 4, let's see. Four is linked with 1 and 5. So 1 and 5 is linked. My last row, number 5 is linked with 1 and 4. Okay. So 1 second. 1 to 4. Well, we can make changes, so it doesn't matter. So at least I have a matrix looks like this. Is this correct? I don't know. This should be symmetric. So which one? Probably this is wrong. So number 3 should link with 1. So this place should be 1. Otherwise, this is wrong. So this place should be 0 because for the diagonal position, I always have 0. So I need to replace 3, 3, A3, 3 with 1, with 0. And I need to replace 3, 1 into 1. Okay. So I make a correction and hopefully this is a network. This should be the network, this. Are you okay with it? So this is just one way to prepare a network. Another easy way should be you should have a description of the links. If you have a table, looks like this. If you have a table, say, instead of directly type this, you may have a table, say 1, 1, 3, and it's linked, so it's 1. I can see 1, 4, it's 1. 1, 5, it's 1. So I can have a table, looks like this, right? So this is another way to do that as input to prepare your network. So there are 2 different ways. One is directly give this matrix, adjacency matrix. Another is you prepare a table and put the node 1, 3, which linked here, 1, 4 linked here, so this is like a linked table, linker table. I don't know whether this is good. You prepare linker table, so there's different ways to prepare. Now we just assume you already have this A, so A is defined. So what's next? I will check whether A is symmetric. So this should be a function in MATLAB. Yeah, so this is a very basic function. I check whether A is symmetric. It is. So looks trivial, and because in this small example you can see it, but for the larger one, you want to check whether your input, the adjacent matrix, is valid. So what's valid? Because we are talking about an undirected graph, so no direction. So you expect the link between 1, 3, 3, 1 should be the same. So you have to be symmetric. So we test this is okay. So now we can prepare the graph. the function of graph is just graph. In MATLAB, you gave the A, and the graph will be created. So graph is a function. Will take your input in this way, or I can show you later how can you give input using this table. So the graph, this function will take this as input and prepare the network for you. It counts how many nodes, how many edges in your graph. So to prepare this table. So this is like edge table, this node table. So that's it. So then what you can do is just plot G. That's it. You should see this graph, and you can compare this with your original. So topology is correct. So let's see. The label is not label 1. Yeah. Right? So this just layout different because there's only a single link, so you can put 3 somewhere. Yeah. So this is the network. Okay, so next we have this graph. Once we have this in the memory, in the workspace, we can do a lot with this variable or this object. So you try the plot. You can also use plot as in this way. So this is called so the same thing, okay? Instead of call a function, you can call this as a function associated with this object. You can have other properties. For example, if you use this graph dot adjacency, so this will return the A matrix back. So this G is a variable, hold that information. That adjacent matrix is here, okay? But when you print it out, it gave you a sparse matrix. Sparse matrix, so you see this B, you gave the B. B and A are the same. The only difference is internally in the memory present differently, and this will be more effective in terms of space occupation. The sparse matrix occupied a smaller footprint in the memory. The reason is most of this network are sparse. There are a lot of zeros in the matrix. Okay, so this one, you certainly can obtain the full matrix by using a function and transform the sparse matrix back to the full matrix. Now A and B should be the same. So this is a B. You can start to visualize, or you can double click your original. They are the same. So what else you can do? You can do a degree. Okay, what's degree? So degree is just for each node, how many direct linked number of nodes as neighbors. So the first node should have a degree 1, 2, 3. The second node should have degree 1. And third node should have 2, right? So when you call degree and as a function, you should get an answer which these are the degree for each corresponding nodes in the network. Okay. Remember, we have learned a couple of centrality measures. So now you can directly obtain the centralities. What we had, we mentioned the closeness centrality, right, in the class. So you can just directly compute as a property for this graph and get the centrality, closeness centrality from the graph for all the nodes, okay, directly. If you don't remember which centrality we can use, remember you can tab the tab and to get this suggestions. So those are the valid centrality we can compute directly right now without helping from any other external functions, because this has been built in as a function, which internal function for the graph. Eigenvector. Page rank. Remember the first class we talked about the page rank. It's one of the centrality measures you can use. You can also type dot and then tab. You can check whether there are other interesting functions you want to use. There's some basic information about how many, the number of edges, number of nodes. You can manipulate the graph by remove one of the nodes. If you decide I want to change the graph, delete the edges or nodes. You can also see I want to do the shortest parts between from number 1 to 2. So 1 to 2, and this will by 2 steps. So the shortest parts will go through number 1 to number 3. So this function calculate the shortest parts from node 1 to 2. How can you go? You go from 1 through 3 and to 2. So to highlight the possible paths from one place, one node to another. If you do, that's 5 to 3. If you go 5 to 3, the suggested path is 5, 1, 3. So that's from here, 1, 3. Okay. So this is just a very small example. When you have a large network, many things you cannot easily by just looking, and you have to rely on those computation and to see, I want to find the top 5 nodes in the network give me the largest closeness centrality, for example. Those kind of questions you can ask. So you have this idea what to do once you have the graph. Let me just quickly show you in the toolbox how we can construct a graph. So in this example, let's load any example. So the first function I want to show you is under the menu of network. The second option, this option is independent from your data. So this allows you to put a linker table, and we will generate the network for you. So it's convenient for you if you don't have idea how to type this in, how to prepare this input using command line, but here you just directly create those. So you can see I want to paste the text. So the example will be something like this. So this is a linker table, and I use gene name as example. So you have each gene is one of the nodes in the network. If this gene is linked with this, and you make a link, so each column is not a unique set of genes. You see the repeats, right? Because one node may have multiple linkers around it. So depends on your application, whatever you get something like this list, you can type this in here. Then just click OK. The program will prepare this network for you. Remember, this is, you see the arrows, okay? So this is a directed graph. The reason is this link table, I put 1, 3, but I didn't put 3 to 1, okay? So this is not a symmetric. This one gene regulate another gene. They have direction, so you see the arrows. So the first thing first, if you decide to export this, you can save the network into as a graph. You can export the adjacency matrix. You can export the graph, and you can save the name of variables, which is a gene list. Okay. So that's what I replace the workspace variable. Now my graph, my adjacency matrix is 26 by 26. And I have a list of genes, which 26 genes. Adjacency matrix look like this. It's not symmetric. If you do this, you will see it's not symmetric because the network we build from the adjacent matrix is directed network, so not symmetric. And you have a graph. So the graph, the properties, nodes, not number edges, you still can do the degree and as you did before, okay? So you have to so-called in degree. The reason is this graph is directed, so you have to consider degree whether this link is go outside from the node or go inside. So you can compute the in degree or outer degree differently. Okay, so come back to this. I can show you what we can do next. So this nodes, you can change the font size of those labels. You can drag, you can change the position to move, to make this okay. So you can move them. So anytime you can say, I want to export this into PowerPoint. Once you are ready, okay, your network is fine. You should be able to click the button to export this into PowerPoint. Yep.
So if you only wanted to see the network between, like, let's say, like 3 or 4 genes,
Mm-hmm.
like in reality they have nothing to do with each other, like like a biological
Mm-hmm.
way, would it still show you arrows and stuff and like indicate that there's a relationship?
Well, it depends on your input. If you prepare this, you have 5 genes, and if there's nothing linked to each other, you should not give any this network representation from the beginning.
Oh, okay. So
Yeah.
like basically, if there was nothing to be
Linked, you
wouldn't.
wouldn't bother to do this network analysis. Yeah. So if you prepared this, you assume there's some link, directed, undirected, or from observation. You can, so this is the preparation, and there are different ways to change the weight. Okay, you should be able to, but this is not weighted, so you don't see any. You can click this button to export this network into something slightly different. Sometimes this gives you the better view. Okay, basically this label will be put in the middle of the node. In this case, not very good, but sometimes you need output to look like this. You can also select this way to do the output. So similar to previous, but it's just using the link with curved. So and then anytime you can export this to PowerPoint. Okay. With this, you can also, so this is unweighted, so you don't see the change of the link, the weights. But you can use this to change the layout. If you click this, there are different ways to rearrange your nodes. Okay. Occasionally, you want to use a circle, but you can try this other. MATLAB provide internal function to help you to rearrange your nodes. Okay. So I have function to make directed network into undirected. So this will change the property. Command line, as I said, you can still use the command line function to plot a graph. So this will be the core, and you will see the same as we did inside the toolbox. But in this case, you don't have other options that I gave to. You have to copy and paste into the PowerPoint, for example. You can do centrality. You start to see more. In this case, you have in closeness, in degree, outer closeness, outer degree. The reason is those centrality, and consider whether the direction of links can compute between this centrality. Now we can look at the documentation for the graph and see what other functions we can utilize when we have a graph. So you can give the nodes different names. Right now, I just gave, if you don't give anything, just directly, just this, the nodes will be labeled as 1, 2, 3, depends on the rows or columns. But if you want to name, say, node number 1 is A with a string, then you can manipulate the properties. You can add 1 and add H. So they're all examples of those. When you plot a graph, you can see I want to label the edges, their edges, by some of the information of what you want to label on them. In this case, I want to label the edge's weight, and then the shoes. You can so this weight, and 10, create a weight graph using a list of the end nodes each. So that's add. You can start adding edges. You can start by creating an empty graph, then gradually add edges in there. You can start from a table and the nodes. There are many options. Let's look at this graph and network examples. So you see there are different ways to annotate your graph with a color, label different colors with for the nodes and the linkers as well. Many options. I think this will be easier. Let's try use a page ranker to give weight for a network, which contains the nodes. So let's try this example together. So we move down, we copy this piece of code. Let's just build this. So this network is weighted and directed. So you see the errors from one node to another, and also there's a number on it. So those are the weights. This is another example with 6 different websites. Copy and paste code. And copy this plotting code. So that's this, and then we compute the page ranker values by call this function, and then the page ranker PR values for each node been computed. And then I think the last one we will rank this, see which page is the most important. Okay, so let's see the time. So now you know, once you have this or have this, what to do. So you can create graph. Now I'm going to show you, if we have this expression matrix, how can we get the network itself, right? Now we can close them, close all of these examples. Clear. Come back our application. Load example data. So our example data have multiple cell types. So you don't want to have a network contains different numbers of different cell types. We want to build cell type specific network. So we are going to extract only one cell type from this data and prepare the gene sets to build the network. So what you can do is to select one of this group, select by cell type. Then we will select alpha cell, okay? Alpha cell. close the original. So we only work with alpha cell. So we have 612 alpha cells. We are going to build a network for them. There looks like there are two subtypes or subgroup of alpha cell. So if you want to do more specifically, so you can select only work on you can only work on those or that to build two networks for each of them separately. Because they from a different, I believe they from a different time. Yeah. So. Yeah, I think we can do 300 cells, maybe enough. Yeah, so I think I will just brush and get rid, then delete. So delete those, select the cells. Now, there are a couple of them from the different samples, but that's okay. So I'm going to build a network for this 286 cells. Okay, now there are too many genes, so I want to do a filter by, if you look at the network, I remove these informative genes. Okay, you click this, and this is the mouse data. So there are many options here. For the last one, maybe you only want to keep 2,000 genes. All this. Let's see, 1,500. I don't know whether after this filter we will still have enough cells. If not, we have to include more cells to do, but just give it a try. So this will remove 206 cells, so many genes. I don't think we can do it because there are only 6 cells left, 80 cells left. There's too few to build a network. So I think we can compromise some of those criteria. So ideally, we should have a very clean, homogeneous, relatively homogeneous population of cells in order to build a network. But in this example, when we extract cells which satisfy this condition, very few cells left. So we need compromise some of those criteria to select more cells. So I'm going to build a network for all of them, whatever. It's not realistic, but just do this, remove to build a small network, only contain 1,500 cells by removing. So now I have been left with this matrix with 1,500 genes, 775 cells. And we can export this data into MATLAB workspace by click X and gene. Okay, now I have prepared my X matrix with reasonable number of genes, so we can show the network, and won't take too long to build. And so that's our input. The number of cells is 775. Okay, So once you finish this step, we can take a break, come back at 11 to move forward to build the network. Anyone have any questions? Yeah, okay. just have to learn how to construct the network from the expression matrix. The first thing I want to do is to manipulate, impute the matrix. So you remember magic, okay? We use magic to impute this X matrix. So now because this X matrix contains so many zeros, but after imputation, this XM version will be more suitable for correlation analysis. Easy, right? So just one step. Let me show you again. So this, do the imputation. The next step, we are going to construct co-expression matrix. So the idea is if you use correlation and then get the first gene, remember the rows are the genes, and XM, the second gene, you will have a, let's see, transpose it. You will have a number. This number tell you number gene 1 and gene 2 highly correlated there. Have a correlation coefficient 0.84. And if you change this to gene 3, this number will change even higher, and then number 4 lower. So we can try this with any pairs of genes to get the correlation between genes, right? So you can do this for all the pairs. So if you gave the matrix, this should give you the covariance matrix, correlation coefficient matrix. So you don't have to do one gene by another. So you just do the whole group and put that in the function. You will get this R, which is the correlation matrix across all the pairs of genes. So we have 1,500 genes in this imputed version. And then our R is 1,500 by 1,500. That's between each pair. So you can use R as your adjacence matrix. Okay. The highest value means they are strongly linked, correlated. And weaker ones, 0.4, some of those 0.3. So gene, for example, this one, that's gene 4 and gene 10. That's not strongly linked compared to the other pairs. So this matrix R can be used as an adjacence matrix. However, because it's fully connected, you will get a ball and every gene connect with others. So we need to set a cutoff. See, we can set up, say, 0.85 above should be the linked, otherwise it should be unlinked. So you can set a cutoff, see, A, B's are, the value are greater than 0.85. 5. So A becomes a 0, 1 matrix. And this is symmetric. So you can, you know how to play once you have A. You know how to play with downstream. So now A is prepared for you. So this is the network. So G is graph from A, right? G node Yeah, I'm thinking whether I should plot this. This is a 1,500 by 1,500. Maybe it's okay. So this is a network, okay? So this network, you can see a lot of loops. This gene by itself, loop to itself. The reason is the input, the diagonal of this position contains 1. Because when you do the correlation, the gene correlates to itself, the correlation coefficient is 1. So when we do the cutoff, this remains as 1. So every gene has its self loop in the network. I think we can get rid of it by doing a very easy transformation. So A is here, so we want to put 0 in the diagonal position, right? You can do it by A minus. Okay. So this transformation will remove that. Now if you look at all the 1, 1, 2, 2, 3, 3 position all become 0. And now we can reconstruct the graph using this new one and the figure to plot. Okay, now you have built a gene regulatory network or co-expression network, basically. This is not considering any negative correlation, and because we take the absolute values. There are some other manipulation you can do if you consider the negative ones differently. But for the simple operations, this basically it. With this, you can compute which gene have a highest degree. For example, if you do the degree, so then we can do a sort. You sort the degree by the descending. Okay. So I don't need the sorted results. I just need the index. So which one will be on the top. Now I have a gene. Remember, we have a gene. So your index number 1. So this will be the gene have a highest degree in the network. The gene is called MYL12B. Okay, now for this graph, let's give the output into a variable. So before I just plot, and now I decide to give this plot, the results, into a variable called p. The reason is I want to manipulate this figure. I want to do something with this p. So this p, you can change the dot and then say layout, and you select different ways to do the layout. Subspace, for example. If you do this, you select different ways of layout. You should be able to change the display of this. Okay. So the code here is I keep the figure, the plot results into a variable p, and then the p is manipulatable. So I can change layouts, I can change other related with this figure. If you do circle, this should be give you a circle, because all the nodes is placed in this equally distributed on this circle. Well, this takes longer than I expected. I don't know why this layered layout will take so long to compute the position of those nodes. Yeah, so I may have to restart my machine, and then this take too long. Okay, I stopped this process because this layout takes too long to finish. But that's just gave you some idea then what you can do. You also found some nodes, they are isolated from the whole network. There are no partners, no linkers. You may want to treat them or remove them from the network. when you do some of the analysis, not allow you to have those. Anyway, so this is from X matrix to build a covariance co-expression network and set cutoff, and then build a network for yourself. Now we are going to come back to the two interface here, okay? In this case, we are going to build a smaller network with selected genes. So if you select this item under the network menu, this allows you to select a set of genes. If you don't select, I will give you a random set of genes from your data. But if you know which pathway or which gene set function you want to get those genes in there, you can type those in. Then you click this, you have two options. PC regression stands for principal components regression, and the Chatterjee correlation. Remember I told you about the Chatterjee correlation, which is a new correlation coefficient different from the Pearson and Spearman and recently proposed. So you can use either of them to build a network for the gene you selected. We can do one of the transformation. You will get something look like this. So this looks different from what we described starting from here. The reason is this is a directed, undirected, symmetric, and also not weighted. Well, this network is directed, so you have a direction. And also blue indicate positive direction, positive link and the correlation, and this orange or red indicate negative correlation. Yeah, quite. Oh, how to start from? Get this?
How to start the previous process?
Oh, I just use Control-C, and I tried the Control-Shift deletion, so I tried a couple of things. Let me try like this. Yeah, let's show that again and show you how you get this. So this is for small network. So you have to have some idea which GA you want to include in there. Not like we build a whole network and you don't know what to do. So this is a so-called local. So you select a set of genes. So that's in the network, and build gene network using selected genes. you select or you paste list of genes here, and then using one of the method. To get this. So each time I have this select gene changed, so your network may not same as what I have here, even the number of genes. Okay, so certainly you can change the font size to see them. And you see one of the genes is highlighted as a red here, KLF7. So this is a transcription factor in the database people knows. Okay, so if it is a transcription factor, I will highlight it into red. So blue line indicate this gene and this gene, they are positively correlated. If this gene high, that gene will also high in the cell. The red line indicate they are negative correlated. So if you have a negative correlation between this and that, two genes, the link will be in orange. And some link looks thicker than another, so that's because these are the directed and also weighted. So you can change the layout. This is a small network, so I can do a lot of things. Okay, we can set a cutoff. There are too many links, so I can set up, say, I only need 75% of links. So the links with weak strings will be removed. So if I set cutoff, it will show a more sparse or weakly linked network. This may make more sense to you. The genes in the center strongly linked with others still have a lot of links. Okay. So you see they are strong link, then you have a thick, the wide this linker. If you change the view, you can see this is stronger link, which this link is weaker. The thinner the links. This algorithm basically gives similar weight between two different directions, so you don't have to use a directed graph. You can change this to the undirected. Trim off. You get something like this. Change the view. Change the font size until you found it's useful. Then then export this into different views and save this. This is fully connected, still the older version. So different views. There's a movie, you can click this. For example, you don't know which cutoff you want to select. You can start from the fully connected, and then the algorithm will remove step by step. If you think, okay, now we can stop, you just click cancel. The this cutoff will just stop here, and then you get this certain level of sparsity from the network. You stop there, and then I don't know whether the underlying network still not change, but there's a way you can change the underlying network after the trim. So if you don't keep original, then I hope that you should change the underlying the graph. For this example, the genes are randomly selected, and the cell population doesn't make sense because it contains a lot of different cell types. So your own research should be cell type specific, and you select targeted gene sites you want to play, and then you know which one should be positively regulated with others. So that, in that case, may give you some good idea about your systems. But here just show you the possibilities. You can use those to do those things. These basic functions. So the next item in this network manual is to build gene regulatory networks and also compare. So this function is similar to the previous one. You will have a chance to select the gene sites. But before that, it allows you to select two groups of cells. So consider now in your sample, you have two groups of cells from two different conditions. In my case, it's week 8 compared to week 14. Then you select the genes, same set of genes for two sets of cells in different groups, treatment groups. Now you have two networks side by side. So left is your week 8 versus right side is week 14 network. So you will see slightly different correlation values, assuming these two time points, the connections shifted or strength changed. So you want to visualize this difference, also set up the cutoff. See, I want to set up cutoff at position 0.3. Yeah, maybe 0.4. So yeah, sometimes something like 0.4. So I keep 0.4 as a cutoff, and then change to undirected, and change the layout. And the hope is I can identify the place where these two networks differ from each other. This the example here is not very good because I select a random set of genes. You don't expect to have strong effects change. So it looks very similar to each other. But if you select the right conditions and the right set of genes, you may see the change of those links. Okay, so any questions for this part? Yeah, so I mentioned the single cell Tendernet during the lecture. So this will be used to construct and compare networks and find a gene, which gene have a major regulation partnership change of their to identify those. So you can access these functions through this menu here, okay? So the gene constructed, you can construct this whole 1,500. So in this case, you don't select gene, you just construct the whole global network for all the genes. If you decide to compare, you can select this. If you don't do the subsampling, so you don't do this time-consuming steps. You just directly jump from A to D. You can do this by select no, because there's a rabbit, and this algorithm will be faster. You can select this and tell you where the reference is. So this allow you to save intermediate steps. Now we select two groups of cells, okay? Because we are comparing two sets. So this will start to build two networks, the whole 1,500 against another 1,500. So the program is working on that. You can see the intermediate steps. And also invoke this parallel computing power, because you have multiple core in your PCU. So this algorithm will reach out each individual cores to break down your construction of network task into small tasks, subtasks, and run them at the same time. So that will improve the speed a little bit. And then I hope we can finish this in 1 or 2 minutes, something like that. So do the comparison. So yeah. The output we have to wait. The output will be a list of genes. Which you got the output already? Your computer is super fast because you use a smaller gene set. Yes, that's good. So you have output. Yeah, so you have a smaller, yeah. So the top genes will be the. No, it's already yeah, it's sorted. It's sorted by the significance. The top genes will be the change. Yeah. Yeah, that's it. Yeah. So the output will be in the folder you selected. In the middle of you prepare this, and then the result will be an Excel table, and the table contains a list of genes. On the top ones are those major difference between these two samples in terms of their gene regulatory network, topological relationship with others in the strains. It's constructing the second network. So we need two networks. The first one already done. The second one is ongoing. And after finished, the last step, we will just compare them and give you results. So the network construction is a time-consuming step. Now to the manifold alignment, it'd be very easy, and then we finished in, it's already done. So you see, okay, it's in this folder, the results, the output is here. So you can double click and open this Excel spreadsheet. So this gene list have been sorted by this p-value or the distance, whatever you call it. So in my comparison of these two networks and this gene on the top, what that means, their regulation relationship with other genes changed from one network to another, changed the most. Okay, have you got the results yet? No, still wait. So you have some idea if you use only 1,000 genes, it'd be very quick, 2 or 3 minutes. We have 1,500 genes. Took me about less than 10 minutes, okay? But if you have 5,000 by 5,000, you may have to wait for 6 or 7 hours. You can still try 8,000 by 8,000. I don't recommend to go further because that takes too long. So the next one, what you can try is to do this simulation gene knockout. So you can just follow instruction. In the middle of this algorithm, you have to select a gene which you want to knock out from the sample to simulate its impact on the network. And the next one will be a similar function, but it requires you to have the Python installed, okay? Then you have cell-cell communication. The package also requires Python, so you have to install Python and then prepare your data here and send it to the Python through this interface to get back results. And the last one, we are still working on it. Sharon is working on that project to simulate you delete the interaction links between two cell types. see whether you can change the crosstalk between different cell types. Yeah, you should construct de novo because you don't have a previously constructed. If you want to do, for example, you delete one gene, but next time you don't have to reconstruct the network. You can borrow the previous network you constructed and try to delete another and see what happens. Yeah. But the first time you have to construct the network yourself. Okay, that's about it. And next week, you just stay home and prepare your presentation. Let me know if you have any questions in the middle. We don't have to come back to the classroom and until May 6th. So this will be the week after next week. Okay?